Hello friends and dear students. Today I am going to discuss on a worksheet on plant tissue culture. A visual question may be asked in your CBSE exam or in your competitive exam. So first of all, try to identify the figure depicted in this diagram, that is in plant tissue culture, that is if you see in the petri dish that contain media and the small section of the plant leaf, that is X plant. So this is X plant through which we can initiate the plant tissue culture experiment. Next, second is identify the stage of plant tissue culture in the following diagram. This is callus, callus, undifferentiated growth of plant X plant part in plant tissue culture media having equal amount of oxygen and cytokine. So this is the X plant that is going to derive from, that is this is the callus that is derived from X plant. So this is the second steps you get after growing the X plant, that is the callus. You cannot define whether it is the root part or a stem part. If there is a equal amount of oxygen and cytokinin concentration, then you get the X plant, because oxygen and cytokinin are the plant growth regulators, and if there is equal amount, then it will grow as undifferentiated, means you cannot say it is the root or it is the stem. Next diagram, that is identify the diagram related to plant tissue culture, that is artificial seed. Artificial seed, that is very helpful in adverse environmental condition when there is all the plantlet or seedling are disturbed or ruined due to environmental condition. In that situation, you can quickly grow the plant from the artificial seed, where somatic embryo encapsulated with calcium alginate or sodium alginates. So this is a diagram related to uh, artificial seed. Next question may be asked, explain the process depicted in the following diagrams. So it is also a steps of plant tissue culture. Generally, if you see from the X plant, you can develop the callus. From the callus, you develop the stem part and then root part. So till now, there is no proper development of xylem and phloem. So this is the process of hardening, means development of xylem and phloem inside the newly growing plant, inside the plant tissue culture lab. So you have to put in glasshouse or greenhouse. That process is known as hardening, means development of xylem and phloem to make the plant autotrophs, because in earlier it is taking nutrients from the media. Now next, identify the worm related to BT cotton present in the following diagram. Cotton bollworm or pink bollworm that disturb the balls. That ball contain fibers, cotton balls. So that is responsible for degrading the fiber present inside the ball. So that is either cotton bollworm or pink bollworm. So scientists have prepared the BT cotton to check such type of damage by that uh, cotton bollworm or pink bollworm. So try to identify. If question may be asked in your CBSE exam or in your competitive exam, identify this. So you have to identify, because uh, it is going to affect the production of cotton fibers. Okay, so a BT gene present in the plants of cotton that create a BT crystal protein when it will taken by this cotton bollworm or pink bollworm. This is in liquid form, as as this liquid form of BT gene crystal protein enter in the gut of this cotton bollworm or pink bollworm, it becomes solid crystals, and that rupture the crop or intestine, and that damage the cotton bollworm. Next, identify the techniques in the following diagram for the production of flavor saver variety of tomato. Why it is advantageous? You know, antisense RNA technology, that is hybridization of sense strand of RNA, that is responsible for ripening of the fruits by antisense strand, that will prevent the formation of enzyme responsible for ethylene productions. So delay ripening of the fruit occur due to that antisense RNA technology inside the tomato. So we can increase the shelf life of the plants. So that variety is known as flavor saver variety, and we can increase the shelf life of that tomato variety. Write down answer of the following diagram. You know, in plant tissue culture, you can identify that is the normal rice and that is golden iron rice. That normal rice do not contain a gene responsible for beta carotene or irons. So if a person suffering from vitamin A deficiency may suffer from night blindness, and uh, children or men or women suffer from anemia due to deficiency of iron. Okay, so advantage of that golden iron rice, that contain beta carotene and the iron, golden iron rice. Okay, so the three gene, PSY gene, CRT1 gene, CRT2 gene, that entered inside the golden iron rice, responsible for beta carotene production. And the next diagram, that is globules of polyhydroxybutyrate, that is responsible for natural plastics, that is biodegradable plastic, that is going to develop inside the aerobic systems. So you have to identify the diagram also. It may come in your competitive exam or in your CBSE exam. Now come to write down the source, number of the genes for the production of golden iron rice in the following diagram. You know, daffodils of flower, that provide the gene PSY gene, that is phytin synthase genes, and the two single bacterium gene perform both the functions, Erevenia keratopora, then phytin uh, carotene CRT1 gene, and lycopene beta cyclase. So these are the three genes that is going to transfer just to make a beta carotenes. So you have to understand the what are the total number of them. That is the three, that is PSY gene, CRT1 gene, and CRT2 gene. Okay, and that was taken by daffodils and bacteria. Next, you know, beta carotene is a precursor of vitamin A, due to which there is no chance of night blindness. Your eye will be normal because it will help in visions. Identify the molecular technique for the production of flavor saver variety of tomato. You know, I have already discussed antisense RNA technology. Okay, so normal mRNA, that is messenger RNA, that code for polyglucoronase enzyme responsible for ethylene production. So scientists have inserted the antisense complementary RNA against sense RNA, and due to complementary mRNA hybrid duplex formation, no synthesis of polyglucoronase gene enzyme occurs, and no ripening of the fruit occurred quickly as per in the normal tomato. So antisense RNA technology is the technique through which we can prevent the quick ripening of the tomatoes. Okay, so antisense gene that hybridized with the sense gene that is responsible for polygalactonase enzyme that is responsible for ethylene productions. Next, identify the process in plant tissue culture for the production of hybrid seed. You know, regeneration, promoter with induced transcription in male reproductive, specifically gene which disrupt the normal function of the gene. Agrobacterium mediated gene transfer techniques. So here is BARNASE bar star systems that induce for the creation of genetic male sterile plant, that is female plants. Okay. Next, which mechanism is going to explain the following diagrams? You know, BT gene transfer, the BT gene that encode the BT crystal protein inside the plants. Although that gene encode the BT crystal protein in the plant in the liquid form, as the BT cotton bollworm suck the liquid juice of that plant. When that juice containing BT crystal protein that is in liquid form enter in the midgut of the that insect, that is cotton bollworm or pink bollworm, it becomes solid crystals, and that disrupt the intestine or crop membranes, and that in this way that is going to kill the uh, cotton bollworm or pink bollworm. So in this way we, we can protect the product plants, cotton plants or any plants that going to affected by cotton bollworm or pink bollworm, and enhance the production of the cottons. Okay, so what is the name and the source of the promoter in the following diagram for induced genetic male sterility? You know, agrobacterium mediated gene transfer technique through which we can cauliflower mosaic virus CM35 promoters, and we get the genes from the tobacco plants. For the transfer of the gene to make the pollen grain sterile and make the female plants, just to develop the hybrid through BARNASE bar star systems. So cauliflower mosaic virus CM35 promoters induce the transcription, help in process of functioning of the BARNASE gene that make the pollen grain sterile, and bar star gene that restore the pollen grain fertilities. So that is our BARNASE bar star system only functional with the strong promoter, that is CM35s. Okay, so such type of uh, BARNASE bar star system is very helpful for the production of a good variety of hybrid seed through creating male sterile line. Okay, so BARNASE gene is cytotoxic gene, cytotoxic, killing the tapetal cells, thus prevent the pollen grain development, result in the transgenic male sterility. CM35s promoters. Next, you know, yes, which chemical is used as a cryoprotective agent for putting the plant sample storing in the following process? That is DMSO, dimethyl sulfoxide. In this liquid we can put the sample in the cryo container that contain sufficient amount of liquid nitrogen. In at very low temperature it is not going to form the ice crystal, so we can store the sample for a very long period of time, and it cannot damage the cell structure, maintain the cell in live conditions. Next and last question, that is the what is difference between in situ and ex-situ conservation on the following diagram. If you are growing any plant samples or plants or any storing any sample in lab condition or in protected conditions, then we can say XII conservation, means glasshouse or greenhouse. But if you are maintaining or growing the plants in natural environment condition, then in situ conservation. So these are some of the question may ask in your coming CVS exam for biotechnology, or in your so many competitive exam. Try to understand the term, that is ex situ or in situ. Other similar word is in vitro or in vivo. In vivo means lab conditions in presence utilizing any living organism for your any experimental purpose. In vivo or in vitro means lab condition, but you are not going to utilize any living organism, that is known as in vitro. So try to understand the concept of biotechnology terminology related to biology and proceed for your coming exam. Thank you and have a nice day.